Good afternoon, everybody, and welcome to the Brain and Behavior Research Foundation's Meet the Scientist monthly webinar series. I'm Dr. Peter Tarr, and I'm BBRF's editorial director and chief science writer. And I'm sitting in today for Dr. Jeffrey Borenstein, the regular host of these webinars, who had an important commitment elsewhere today. In this afternoon's webinar, Dr. Avishek Adhikari will present neural circuits controlling food seeking. Before we do that, I want to just remind you that BBRF is the world's largest private funder of mental health research grants, supporting transformative discoveries in order to develop improved treatments, cures, and methods of prevention. And the high quality of the research that we fund is made possible by BBRF's Scientific Council. This is a group of 191 prominent mental health researchers who review each grant application and select the most promising ideas with the greatest potential to lead to breakthroughs. The Scientific Council guides the foundation to fund creative and impactful basic, translational, and clinical research relevant to the whole spectrum of mental health. One reason that the research that BBRF funds has such great impact is because we do not limit our focus to one illness or condition. Since 1987, the foundation has awarded $476 million to fund more than 6,800 research grants around the world and across a broad spectrum of brain illnesses. And these include addiction, ADHD, anxiety, autism, bipolar disorder, borderline personality disorder, depression, eating disorders, OCD, PTSD, schizophrenia, as well as suicide prevention. Today, I'm delighted to introduce Dr. Avishek Adhikari. Avishek calls himself Avi, and I will call him Avi, if you will permit me to, in this presentation. Dr. Adhikari is an associate professor and the Wenzel and Jeffrey Chair in Behavioral Neuroscience in the Department of Psychology at the University of California, Los Angeles. He received a BBRF Young Investigator grant in the year 2014. Our webinar will begin with Dr. Adhikari's presentation, which will then be followed by a Q&A. So to submit your questions as you're listening, please use the questions tab on the control panel on your screen, and please feel free to submit questions at any time. And following Dr. Adhikari's presentation, I'll ask as many of your questions as possible in the time allotted. So for now, I'm pleased to introduce Dr. Adhikari. Please take it away.
Well, thank you, Peter, for that wonderful introduction. And thank you all for your time for listening to our research. And thank you also for the foundation for supporting our research for all these years. So we'll start with the talk now. And today's talk is on how the brain controls food seeking, which is a fundamental behavior for survival. And then when food seeking circuits in the brain go awry, it can cause both pathological decreases in eating, such as anorexia, or also increases in eating, such as in binge eating disorder. And our lab historically has not really studied food seeking and eating, so this story has sort of a unusual path. And most talks that you might see in science have a usual structure that goes like this, where the speaker might say that based on these prior data, we predict these super specific experimental outcomes. And then we observe all our outcomes. We are so good at predicting things. We should be on Wall Street instead of doing science. But then our talk structure will be very different because it came from a surprising result. So our talk structure is, so based on prior data, I made some predictions. Then we didn't observe any of the predictions. So we're very confused and sad. But then we saw some unexpected, interesting results. And following up on those results is where we get the story from today's. And our conclusion is that we are very bad at predicting, but with unexpected results, we can find new discoveries that no one was thinking of looking at before. Okay, so our story focuses on this brain region called the periaqueductal gray, or PAG, which both humans and mice and many other animals share. So here we have a brain image showing the location of the periaqueductal gray in the brainstem in humans. So this little yellow area here. And this region has been studied for many decades in fear, which is what our lab was interested in. because it was known that if you electrically stimulate this periaqueductal gray region, it can create a panic attack instantly in a otherwise happy, healthy person. So if we were to stimulate this periaqueductal gray region in you, even though you're happy now, you would instantly develop a full-blown panic attack. So very remarkable, strong effect. So we naturally were interested in this region because it's important for fear. And in mice, a similar result was also observed. So here we'll see an experiment from another group showing what happens if you artificially activate the excitatory cells in the periaqueductal gray region in mice. So before looking at the video, a quick kind of introduction, which is in every brain region, there are sort of two flavors of neurons. So there are these excitatory neurons, which you could think of as sort of the gas pedal on the neuron on that brain region, and activating those neurons increases that brain region's electrical activity. So those are the excitatory neurons. And we also have the converse inhibitory neurons, which are the brake pedal in that region. So when they are more active, they decrease that brain region's electrical activity and decrease its function. So here we have a video showing in mice what happens if we electrically activate the excitatory cells in this periaqueductal gray brain region. So what will happen is, at baseline, the mouse is happy looking at things and exploring and moving around and smelling. And then when this brain region's excitatory cells are activated, you'll see that the mouse will rapidly transition into a freezing behavior. And they freeze to indicate that they're scared. And the idea is if they freeze and hide, then they can sort of evade visual detection from some predator, right? So if there was some predator looking for me and I was just moving around like this, it's very easy to see me. But if I freeze and hide in the corner, then it's harder to see me. And then that's what the mouse will do when this brain region's excitatory cells are activated. So first, mouse is just moving around and being happy and exploring. Then when this brain region's excitatory cells are activated, now, when it says light on, you see the mouse immediately stops moving and freezes. So it kind of reacts as if there's some predator to hide from, but there's nothing there. So this is a reaction similar to the human when stimulation of this region, the human causes a panic attack. Stimulation of this excitatory cells in this region, the mouse causes this fear-related freezing behavior to kind of hide from threats. So here you see the mouse is freezing again when this region is being activated. Okay, so the prior work shows both in humans and in mice that when this region is activated, it induces fear and panic attacks and general stress reactions. So we had a very reasonable hypothesis that if we activate the inhibitory cells in this periaqueductal gray region, or PAG, which are the brake pedals in this region, then we should see, well, less fear, less panic attack, inhibition of fear. So that was our hypothesis, that activation of these cells would inhibit fear. So first we tried to see, are these cells more activated during exposure to a live predator, which is a dangerous stimulus for the mouse? And the rat is a predator of the mouse in the wild, which might look strange because it seems similar, but it's not that unusual. So for example, chimpanzees eat smaller monkeys and rat eat mice in the wild. So it's a scary stimulus for the mouse. So we expose the mouse to the rat. So we think that, well, if these inhibitory PAG cells are involved in fear reaction, then they should be activated by the presence of the rat. And here in this heat map, we see the amount of neural activity in these inhibitory cells in each pixel, and red pixels indicate more neural activity. So you see there's some red far away from the rat, some red near the rat. So overall, these brain region inhibitory cells are not more or less active near the rat. So these cells do not seem to be more activated near a predator. So they don't seem to care particularly the presence or absence of this predator, suggesting they are not super concerned with this threat reaction. So that was surprising. Then after that, we try to see what happens if we artificially alter the neural activity of these inhibitory cells in this panic-producing region. So we see that when we expose the mouse to the rat, then neither the control nor the inhibition nor the excitation of these inhibitory cells causes any changes in the escape speed velocity from the rat or in the amount of avoidance the mouse displays to this threat. So it appears that these inhibitory cells don't seem to change the fear reactions of the mouse when it's exposed to a threatening stimulus. So our predictions turned out to be wrong. We don't see that this circuit is inhibiting fear. So then we thought, what else could it be doing? So we looked at it very carefully. We noticed that this region was, when activated, causing sort of a food-seeking reaction in these mice. So the same inhibitory cells in spiracoductal gray region, now when we activate them artificially, they don't seem to inhibit fear, but instead they cause this food-seeking action. So here we have a video of a mouse. So this mouse is not hungry, so he's not interested in eating. And on the left, there's some chow, which is the well-balanced, nutritionally dense food that they eat. And on the right, we have some wonderful, delicious, sweet, milky, fatty chocolate. And at baseline, you'll see the mouse is kind of walking around and it's not super interested in the food because it's not hungry. But when we activate these cells, which you see when the light is flashing, then the mouse goes immediately towards the chocolate and starts eating the chocolate. So this suggests to us two interesting results. So one is that activating these inhibitory cells seems to induce food seeking, and it induces food seeking preferentially towards these more caloric, sweet, fatty foods like the chocolate rather than the chow, which is its more kind of nutritionally balanced food. So then we look more into detail to how this effect was happening, this food seeking effect that was very unexpected. So after that, we try to see what is the neural activity like in these cells while the mice were doing these food eating, foraging related tasks. So the neural activity of each cell will be plotted as a trace like this, as you see on the left. And whenever you see a little peak on the trace, those will be moments in which that inhibitory cell is more electrically activated. And whenever you see these kind of flatter regions of time, those are moments that those neurons are more inhibited or they're less electrically active. Okay, so when we expose the mouse to a walnut, he sometimes eats the walnut, which are these moments in time shown in blue. And each little trace is the neural activity trace of one individual inhibitory cell in the periaqueductal gray, which we called VGAT-PAG neurons. And you see that the VGAT-PAG neurons show more peaks, so more electrical activity when the mouse is looking for the food, which are the white moments in time, and the activity is sort of suppressed in the blue moments in time, so there's more flatness, fewer peaks. So these cells are more active during food seeking, and they're inhibited during the actual eating of the food, which suggests that they might be driving this food seeking behavior. And that suggests that perhaps these cells might be modulated by the hunger state of the mouse, because if they're more active in the food seeking, they should be food seeking more when they're more hungry. So perhaps these cells should be more active when the mouse is more hungry. And then we recorded the same cells in the periaqueductal gray when the mouse was food deprived in orange, and the same cells when they were sated and not hungry. So here you see generally that in the left there are more peaks, suggesting there's more electrical activity of these inhibitory VGAT periaqueductal gray cells. And then when the mouse is satiated and not hungry, there is less activity. So these cells are more active during hunger and they're more active during food seeking compared to eating. And because they're being modulated by hunger, next we thought, well, perhaps these cells might be modulated by a drug that is known to modulate hunger, such as Ozempic. So the active ingredient in Ozempic is this chemical called semaglutide. So that's what we use for this experiment. So first, we just injected the mice with Ozempic, essentially. Then we see that relative to the control mouse in blue, the mice injected with Ozempic eat less. So just like in humans, Ozempic injection in mice makes them eat less. So the protocol is working. Then we try to see what is it that happens with these cells when we inject the mouse with either saline or Ozempic or semaglutide. And then you see that in saline-injected mice, the neurons show lots of little peaks of electrical activity. So these inhibitory cells are active in this sort of hungry mouse treated with saline. But then when we inject semaglutide or Ozempic, the traces look flatter, indicating the cells are less active. These cells are less electrically excited when we inject the mouse with this hunger suppressing drug. So these cells are more active during hunger, and they're also suppressed by this drug that decreases hunger, which makes sense so far. And if we quantify it, we see that there are more cells that are inhibited or negatively modulated compared to cells that are excited by Ozempic. So these cells seem to be listening to the same molecular pathway that is targeted by Ozempic. And because Ozempic controls body weight and it suppresses the activity of these cells, we then next ask, okay, so what would happen if we kind of eliminated these cells from the brain? Okay, so here we have a microscope picture of the periaqueductal gray region in mice. And in green, you can see the location of the inhibitory VGAT cells, which are located in the periaqueductal gray. So there's a lot of these cells here. Then we injected a separate group of mice with a special kind of engineered virus that is able to infect these cells and then eliminate or kill them. So here we have an image showing that if we inject these viruses that are engineered to eliminate these PAGV get cells, indeed we have fewer green cells. So this virus worked to kill these cells. So now this is a mouse that doesn't have these inhibitory VGAT cells in the periaqueductal gray region. So what happens, very surprisingly, is after we make this injection to inject this virus, then this mouse starts losing weight gradually. So eliminating these cells decreases body weight. So it suggests that in a normal mouse, these cells might have something to do with maintaining or gaining normal weight. So it makes sense that these cells are inhibited by Ozempic, which is thought to decrease eating, decrease body weight, etc. And after that, we wanted to see, okay, so what happens if we artificially inhibit the activity of these cells? So we are able to selectively inhibit just the activity of these cells in the periaqueductal gray without inhibiting the activity of any other cell. So we expose the mouse to a food source, which is a walnut, which is a naturally occurring kind of fatty, highly dense food. And these are hungry mice. So the hungry mice are motivated to eat a lot of food and find food and etc. And what we see is that when we inhibit these cells, then mice eat less walnut than they do in the control condition. So even though the mouse is hungry, its normal tendency is to eat a lot of walnut. Inhibiting the activity of these cells makes the hungry mouse eat less. And after that, we did the opposite result, which is, okay, so if we inhibit these cells, mice eat less. So what happens if we activate these cells? So here we'll see that when we activate these cells, it will cause the mouse to hunt this little cricket. So we think of mice as predominantly prey because the cat is eating the mouse, the owl is eating the mouse, everyone's eating the mouse. But the mice are not just victims, they're also little predators, and they predate upon little creatures like insects such as the cricket. So you'll see that at baseline, the mouse has some interest in the cricket. But when we activate these cells, which is when it says on here on the right corner, you'll see the mouse will very rapidly engage and try to catch and eat and hunt the cricket. So immediately when these cells are turned on, you see the mouse's behavior changes qualitatively, and he goes from being sort of interested in the cricket to rapidly hunting and eating the cricket, which is a type of natural food-seeking behavior the mouse has. And if we perform the same experiment with walnuts, we also see that activating these cells causes more eating of walnut compared to the control mice. And it causes a higher amount of cricket eating compared to the control mice. And here, these mice were hungry, so they are naturally motivated to eat crickets and eat walnuts. So even though the the mouse is not hungry. The mouse is not hungry, so they're not super motivated to eat these things. But even though he's not hungry, activating these cells causes the mouse to food seek and eat walnut and eat cricket. So it kind of circumvents the normal satiety that the non-hungry mouse has. And then we wanted to see, okay, so activating these cells can cause mice to food seek and eat crickets and eat walnuts and such, but does activating these cells cause the mouse to be interested in everything, or is it specifically something geared towards food? So we put the mouse in this box here, and then on each opposite corner of the box, we hung either a object that cannot be eaten or a walnut. So the walnut is high on the ceiling, so the mouse cannot reach it. And these mice are again not hungry. So the control mouse spends equal amounts of time in each quadrant, where the warmer colors indicate more time spent in a particular pixel. So the control mouse spends equal times everywhere. But then when we activate artificially these cells, you see that all the red is concentrated directly beneath the walnut. So activating these cells causes the mouse to really pay attention to the walnut and stay underneath the walnut and try to catch it even though it's out of reach. So it causes approach to food, but not other objects. So these cells have something to do with creating this interest, craving, food seeking, but not object seeking. Next, we wanted to see why is it that these cells might be causing this sort of increased eating, increased food seeking reaction. So one possibility is that it causes more hunger, and another possibility is that it causes perhaps more of this craving to get some interesting reward, which would be a positive psychological state. And there's a way in the mouse that you can ask if activating a brain region or brain circuit causes aversion or a positive motivation. And that's shown here in this graph. So basically what happens is the mouse has free access to walk around either in the left or the right compartment. And then you see at baseline, which is the first graph here, the mouse spends equal amounts of time the left and right. So again, red indicates the amount of time spent per pixel. So there's equal amounts of red in the left and the right. So in the baseline, the mouse doesn't have any preference in spending time on the left or the right. But here what happened is, in the experimental condition, whenever the mouse went to the right side, we activated these cells. And then what you see is activation of these cells is sort of liked by the mouse. They spend more time in a compartment paired with this activation of these cells. They sort of like or seek this activation. And because they like or seek this activation, this is something that produces positive motivation. So that strongly suggests these cells are not causing some unpleasant hunger, because that would produce a aversion or negative motivation. So this suggests that these cells are creating food seeking due to some positive rewarding aspect of food seeking, not due to unpleasant aversive hunger. And to further look into that, we try to see, okay, so these cells when activated cause this increased craving towards rewards, then perhaps this increased motivation will be sufficient to make the mouse overcome some aversive negative consequence when foraging. And to do that, we put the mouse in a box where half of the floor was covered in a shock grid and the walnut was placed on top of the shock grid. So in the first part of the experiment, the shock grid is turned off, and then we just see how much walnut does the mouse eat, and then they eat some amount of walnut because the shock grid is turned off. Then the second part of the experiment, the shock grid is turned on, and now to get access to the walnut, the mouse has to endure these very mild shocks. So these are very weak shocks, but it's still aversive. And then you see that the mouse starts eating less because now he has to go through these aversive shocks to eat the walnut. But then if we have shock plus activation of these cells, it rescues the amount of eating the mouse had at baseline. So activating these cells can make the mouse overcome some amount of punishment or shock to get to the food, which shows that these cells can induce compulsive food seeking, which is eating despite a negative consequence, which is something that is seen in binge eating disorder, for example, where people will food seek is despite the negative consequences of excessive eating. So next, we wanted to see what type of food seeking do these cells induce? Do they induce eating of anything or preferentially one food over the other? So first, we just expose the mice to different sources of food, and we see that the neural activity of these cells is modulated a little during eating of broccoli. But you see that when the mouse is eating cheese, the modulation of the neural activity is much bigger, much more pronounced. So these cells are much more kind of concerned with cheese than broccoli, suggesting that these cells might be more related to eating caloric foods than vegetables. And then that's what we try to test next. So we expose the mice first to some nice nutritive vegetables like cherry, bell pepper, carrots, and broccoli. So they're all nutritious like vegetables that are locally grown in California. So the mouse should be very happy. And then if we activate these cells, we see that the mouse does eat more cherry, more carrot, etc. compared to the control animals. But he eats very little, maybe 50 milligrams of each of these foods. But when we activate these cells when the mouse is exposed to caloric foods like sugar, sausage, chocolate, or cheese, we see that the effect is much bigger. So now they eat around maybe 100 milligrams of these highly caloric foods. And again, these mice are not hungry, so they're not particularly interested in eating anything. But activation of these cells causes these non-hungry mice to voraciously eat a lot of these super caloric, dense fatty or sugary foods such as sugar or sausage. So their activation seems to bias the behavior towards eating more of these caloric foods. But here each food was shown one at a time. So then we thought, okay, so what happens if we have the mouse with an option of eating either cheese or broccoli? So the mouse is placed in a maze like this, where there's cheese on one side, broccoli on the other side. Then when we activate these cells, we see that it causes an increase in cheese eating, as we saw before. And then we thought, okay, it probably causes an increase in cheese eating and an increase in broccoli eating. But no, if there's cheese as an option, it actually causes a decrease in broccoli eating. So activating these cells in the presence of some choice of fatty food versus broccoli causes them to decrease their amount of broccoli eaten and increase their amount of cheese eating. So here you can see that the amount of broccoli eating decreases when we activate these cells. So these cells, when activated, cause a shift of the dietary preferences towards more caloric fatty foods like the cheese and away from the broccoli. So it's the opposite of what the doctor would recommend us to do. And then we wonder, okay, why is it the mouse is eating more of this cheese rather than broccoli when we activate it? So we thought that, well, the cheese is at baseline more valuable to the mouse because the cheese is tastier and has more calories than broccoli. So maybe that's why the behavior is directed towards cheese eating. So we wondered, is the reason really that the cheese is more caloric or that the cheese is more valuable at that moment, which are two slightly different things? So to investigate that, we did this procedure where we expose the mouse to cheese and chocolate on day one and see what's the behavior of the mouse in this situation. And then at baseline, they like cheese and chocolate, so presumably they would eat both. Then overnight, we expose the mouse to only cheese. So the mouse ate cheese only for 24 hours. And at that point, the cheese becomes devalued. Because if you're just exposed to steak or pizza for 24 hours and that's all you eat, then you don't want to eat more steak or pizza. You want to eat the other thing. So you can selectively change the value of a food with this procedure. So we see that on day one, at baseline, when we activate these cells, it causes increased eating of cheese and chocolate, as we expected. Then overnight, the mouse was exposed to a lot of cheese. So now the mouse devalues the cheese because he ate so much cheese for 24 hours. And the next day, activating these cells doesn't cause any more eating of cheese, but it still causes eating of chocolate. So these cells don't seem to be exactly causing eating of caloric things. It seems to be causing eating of what's valuable at that moment because the cheese is still valuable on day two, but now the cheese is still caloric on day two, but it's no longer valuable. So the difference is not the cheese is more caloric. The difference is the cheese is less valuable. And it's not something specific about cheese because if we reverse the experiment and devalue the chocolate instead and expose the chocolate overnight to mice, Then on day 2, the mouse eats cheese when we activate these cells, but no chocolate. So it's not that these cells cause eating of cheese or chocolate or caloric things exactly. These cells seem to gear the behavior of the mouse towards eating what's valuable at that moment. And if the cheese or the chocolate are no longer valuable, he'll not eat those things, but will eat something else. And we thought, okay, another way to see if something is valuable is not by its taste, but rather by its kind of nutritional density and the number of calories it has. So for this experiment, we exposed naive mice that has never seen saccharin and then saw what happens when this naive mouse is exposed to saccharin. And saccharin is sweet and tasty, so presumably the mouse would eat that. And then after that, we taught the mouse that saccharin is sweet, but it doesn't have calories. And the way we teach that is you get a hungry mouse and then you give the hungry mouse saccharin. Then the hungry mouse eats saccharin at first, and then he experiences that I was hungry, then I ate a bunch of saccharin, and I continued hungry. So the mouse sort of learns that, well, saccharin is not valuable. Saccharin does not have nutrition, even though it's sweet. And then we expose the mouse saccharin again and activate these cells. Okay, so what we see is to the naive mouse that never ate saccharin, activating these cells causes eating a lot of saccharin because it's sweet. So the mouse probably anticipates saccharin to be very caloric. But then after the mouse learns that saccharin is not caloric, and then we expose the mouse to saccharin, in that condition, activating these cells does not cause any eating of saccharin. So after the mouse learns that saccharin is not nutritious, activating these cells stops making the mouse eating saccharin. So this again shows that these cells are not exactly making the mouse eat sweet foods or caloric foods or cheese or etc. Rather, it's making the mouse eat whatever is considered or anticipated to be valuable. So after the mouse learns that saccharin is not metabolically valuable, these cells, when activated, do not cause seeking of saccharin. And there was one result that always puzzled us, which is that for the naive mouse, when we activate these cells, it seems to increase eating of essentially everything. Like they eat more broccoli, more carrot, more bell pepper, more chocolate, albeit in different amounts. But the activation of these cells never caused the mouse to eat chow, which is its normal food that's in the mouse cage every day. So this is the food that the mouse just eats every day. And if we activate these cells, it doesn't cause more eating of that food. And then we thought, well, maybe it's because the food that the mouse eats every day is kind of permanently low value for the mouse because it just has access to that all the time. So then we thought, okay, how can we make this food more valuable? Well, you can make this food more valuable by depriving the mouse of this food. So normally he eats this chow every day, but then if overnight you remove the chow and just expose the mouse to cheese for 24 hours, then the mouse eats only cheese because that's what's available. And after that, presumably the chow would become valuable again. So this is sort of like if you have a kid at home and just let them eat all the candy on a Halloween day, the next day he will want to eat the not super sweet regular food you have. So that's what you try to do. If the mouse just pumps himself with cheese, then maybe he'll crave this well-balanced, boring food the next day. And that's what we see, that if you revalue the chow by removing it, then activating the cells causes eating of chow, which is something that doesn't happen at baseline. So this too indicates that these cells are not causing eating of sweet things or caloric things or cheese or chocolate. These cells, when activated, are causing eating of whatever is valuable at that moment. And what's valuable at that moment can change from nutritional history or the flavor or the previous availability of that food. Okay, so now get to the conclusions of our work and also some potential consequences for human health. So we showed that these cells are more active during hunger and then they're suppressed by Ozempic, which is a weight loss drugs. And we also showed that eliminating these cells induces weight loss. So those two results taken together suggest that, well, perhaps these pyracdactyl gray V-GAT inhibitory neurons may contribute to the Ozempic-mediated weight loss. Perhaps part of the reason that Ozempic causes weight loss is by suppressing these cells. So that's a potential consequence for human health of these cells that merits future research. Then another result we observed in mice is that activating these cells can cause eating in not hungry mice, which naturally should be eating, and inhibiting these cells can decrease eating in mice that were hungry. So these cells then could be potentially used for treatments, right? So activation of these cells might be used to treat anorexia in human patients because those are people that don't want to eat. So activating them could potentially overcome that not eating motivation and induce more food seeking. And inhibition of these cells might be used to treat potentially binge eating disorder. And other results we've seen is that activating these cells seems to shift the dietary preference towards caloric foods, such as cheese instead of broccoli. And not just that, but it activates the food seeking towards the foods that are more valuable and craved at that moment, based on the mouse's recent history and current caloric demands. So excessive activation of these cells thus might promote overeating of caloric foods that are being craved at that moment. So those are kind of potential consequences of these results for human patients. And to remember, humans also have this periaqueductal gray region, and they also have these VGAT inhibitory cells in the human periaqueductal gray. So these results might be applicable to humans as well. Okay, and before opening up to questions, I'd like to acknowledge the people and entities that supported this research. So these results were primarily done by these lead co-authors, Nitish Patel and Anita Liu, who are graduate students in the lab who are very talented, and also by postdoc Fernando Reis, which was a NARCED recipient twice from the BBRF Foundation. So here we can see a photo of Nitish eating caloric foods, just like the mice we study. And here we see a photo of Fernando and Anita riding a motorcycle indoors, which is not advised for anyone to do. And I'd also like to thank everyone else in the lab that helped with other aspects of the work, to other collaborators at UCLA and NYU that helped with this work as well. And also to my family, my parents Sadhan and Ratna, and my wife Emily that supported me throughout this work as well. And I have no financial disclosures, but I'd like to. I'd like to have financial disclosures. I'd like to have lots of stock options in some mouse cricket hunting company, but so far not yet. And like to acknowledge the help from our funders, which include the National Institute of Mental Health and the NIH, and also the Brain and Behavior Research Foundation, which, counting both myself and the other people involved in the project, funded a total of 4 awards to people involved in this project. And if you look kind of beyond just the people in the project, the people that I trained with were also awardees of the foundation. So I trained at Stanford with Professor Carl Dijsselrath, who received also a NARCED award when he was starting out, and developed this method that we used here to activate and inhibit these cells, which are used now by thousands of neuroscience labs in the world to discover all sorts of things. So you see that this initial support from the foundation really had this big multiplicative effect throughout the world. And my other mentor in graduate school was Professor Josh Gordon, who eventually became the director of the National Institute of Mental Health, who was also a NARCED awardee around the time that I was working with him at Columbia University. So the foundation has been supporting my work and of my kind of predecessor mentors for many years, generating all these and many other important findings. And before opening up to questions, I'd like to just point out that it's very difficult to predict what would be the applications of this work in future research. So there are some examples here to illustrate that. So for example, when people were using perfumes to smell good in front of their other Victorian friends, eventually the same mechanism that sprays perfume was used to spray gasoline in a car and led to the invention of the carburetor in the car. And when Einstein invented his theory of relativity, that theory of relativity is used to calibrate the GPS satellites, which now we use, for example, to order a sandwich through Uber Eats. And in the 1700s, Luigi Galvani found out that if you pass electric current through a dead frog's legs, the frog legs reanimate and twitch for a little while. And this early discovery in electricity led to eventually the development of batteries that we use, for example, in batteries in our iPhone. So a lot of discoveries and applications have completely unpredictable origins, and now we're in the same state, so it's very difficult for us to predict how exactly this application might be used for a treatment in the future, because if these people couldn't predict their future application of their work, we are in no better position than Einstein was to predict their future applications. And then with that in mind, one thing we can say for sure is, although it's difficult to predict what discovery will lead to which application, it is very easy to predict that studying how the universe and nature and biology works is what led to these and all other important discoveries and applications we have in our modern lives. And that will be the same with the discoveries being made currently. So with that, I'd like to open for questions for anyone interested. And thank you again, both for Foundation for hosting this wonderful webinar, and for all of you for attending and for your time.
Dr. Adhikari, thank you so much for that presentation. It was very interesting, and we had many people asking interesting questions, and I thank the people who asked questions during the presentation. Some of them are going to be too technical to present here, because although you might take it up with the doctor at some other time, but for the purposes of our audience, which is mixed, and some people are just lay people without a tremendous amount of knowledge about this topic, I'm going to try to keep the questions general as possible. So, Avi, a number of the questions have to do with all the talking you did about the concept of reward. And I guess one of the things you can say about reward in light of your experiments is that reward in these experiments was kind of a relative thing. It's not an absolute.
Yes.
In other words, reward is not a fixed thing. Reward can be a craving for something can be different under different contextual circumstances. Is that
exactly? Yes.
Yeah. I mean, if you could talk about that for a minute, because it's potentially confusing.
Yes, yes, exactly as Peter said. There are stimuli that we consider rewarding, but that can be very drastically changed depending on the circumstances, right? So in our case here, food would be very rewarding if you're hungry, but right after you ate at the all-you-can-eat buffet, you don't have any more interest in the same rewarding food that you had 1 hour ago. So the value of the reward can dynamically be changing. And whenever the craving is sort of satiated, the rewarding value of that particular stimulus will decrease until you are deprived of something else or have desire for something else. And this is something interesting about these cells, because these cells seem to be gearing the mouse's behavior towards whatever is considered more valuable or more rewarding at that moment, which is different from what is sweeter or more caloric or more novel. Because usually the more caloric thing might be more rewarding, but if you've been overexposed to that, then you now want to eat something else. And that's exactly what these cells seem to be guiding the behavior towards. It's not caloric foods, but rewarding stimuli.
I hear you. So let me ask verbatim a question that came from one audience member. If hunger drive, satiety cues, and body image circuits are inherently vulnerable in some children, what could we have recognized or done earlier in our child who suffers from an eating disorder? And what can future families do to protect a biologically predisposed child before the illness ever takes hold? I mean, is that too far afield from this research to even venture an answer to that question?
Sorry, could you repeat the first part of the question again?
If the hunger drive, satiety cues, and body image circuits are inherently vulnerable in some children, and I guess I should stop at that point and say, is it your understanding that they are perhaps inherently vulnerable in some children, which would underlie some eating disorders?
Oh, yes, that could definitely happen for sure. And sort of every circuit that is in formation in children is more vulnerable than in adults. And the earlier the kind of negative experience happens, the more negative effect it has later in adulthood. And but from this research, yeah, it is somewhat difficult to say what to do with children because first, we're not medical professionals, and then second, this workout wasn't even done in infant mice. These are all adult mice. But research that already exists does show that both eating habits and cultural relations to food seeking and rewards in childhood have a lot of influence later in life for sure. And not just that, even uh, eating and metabolic condition of the mother while pregnant can have lots of long-lasting consequences in the food-seeking behavior of the later adult children.
Let me go on a different track and try to summarize some of what I think you said, and then ask you if I'm on the right track here. I want to sort of reduce and clarify if possible. Uh, so by inhibiting experimentally a certain group of cells within this area called the periaqueductal gray, the PAG, this tiny little area of gray matter that's sort of at the top of the brain stem. So by inhibiting certain cells in the PAG, you can actually increase appetite uh, even when an animal is not hungry. That's so far so correct. Yeah?
Yeah, almost. If you activate, it causes appetite in an animal that's not hungry. And I wouldn't say it's exactly it causes hunger. It causes some kind of increased reward-seeking action. And we think
Well,
it's not causing hunger because the mouse sort of likes this activation. So it's not something aversive like hunger.
I guess I used the wrong word. The words here are very important. I meant to use the word appetite, and appetite is not necessarily the same
That's
thing as
true.
hunger.
Yes,
And
yes,
I meant
but
to use the word appetite.
yes, yes,
So you stimulate these inhibitory cells in the
yes,
PAG,
yes,
and appetite for certain kinds of foods increases even in animals that are not hungry.
yes,
So then you go on to introduce the questions. Does it mean that they're going for, when this happens, are they going for foods that are more caloric, i.e., quote, valuable for survival? But of course, you went on to explain that value in that context does depend on the situation, whether you've been eating a lot of caloric foods or not in the recent past.
yes.
And then the other possibility is that in some way, by stimulating these cells in the PAG, you are in some way affecting a reward response. Yes? Am I right about that? It's about
Yes,
reward, ultimately, so far as you've been able to tell so far.
yes, and after this work, we had done afterwards more experiments in the lab to see precisely that, which um, are these cells gearing the behavior just towards more valuable foods or to more rewarding stimuli in general? And
Yes.
we see that kind of it's the latter actually, because there are also non-food stimuli the mice find rewarding, such as social interactions, for example. And activating these cells increases social interactions, which is a completely different type of reward-seeking behavior. And it also increases seeking of alcohol after mice have learned to binge drink, but not before. So if you get a naive mouse, they're sort of like kids. Kids don't want to drink whiskey. It's very unpleasant. But if people are exposed to alcohol a lot, they eventually learn to like it. And then in mice, we see that a naive mouse, when we activate these cells, it doesn't cause more drinking of alcohol there. But in a mice that have learned to binge drink, then activating these cells causes more alcohol seeking. So it causes, more social interaction seeking, more kind of addictive drug seeking, and more food seeking, all of which point towards exactly what this question was getting at, is that these cells seem to be triggering some broad reward-seeking actions of the most valuable thing at this moment, be that social interaction or cheese.
I'm so glad you put it that way. That really crystallizes it all for me. What's valuable at the moment, as you pointed out when you were talking about cheese versus chocolate, but it really can apply to cocaine or it can apply to
Yes,
alcohol or whatever, which means that this discovery that you made actually has potentially extremely broad implications for addiction as well as eating disorders. Is that right?
yes, potentially, yes. Yeah, this work that we already completed, which is what's shown here, is all focused on food. But the newer results that are emerging from the lab now show that, yeah, these cells are not exactly food-seeking cells. They are seeking valuable rewards at this moment that I'm craving, whatever that might be, kind of a circuit. And you're completely right in your insight. So that means that these cells could be potentially involved in the symptoms of much broader categories of conditions that are unrelated to eating. Like they could be related to depression, for example, where people have less motivation to seek previous things that they found rewarding in general. And those are all new questions that we are currently interested in investigating.
Just quickly, I want to dispose of a couple of questions that I think are pretty important. They should come up in every webinar that involves mouse research. One person said, "Why mice? Why are you doing these experiments in mice?" And of course, part of the answer here has to do with the evolutionary relationship between the mouse brain and the human brain. Yes, please explain that as
Yes,
briefly as possible.
yeah, that's a very good question. And there's many reasons. So the first reason is why can't you do it in humans? And that's because these methods sort of cannot be done in humans because they're very invasive and involve implants inside deep in the brain, and then it's not possible to do it in humans. So that's why, for ethical and medical reasons, you can't do it in humans. But you can still study the brain in other animals that also have brains. And amongst those, the mouse is a very useful model organism for these studies because it is a mammal, just like we are, and they are very small, so they're convenient to house in a lab. Because why not do this in like a rhinoceros? Because it's very big and inconvenient and expensive and rare. So mice are none of those things. And a third reason is that throughout the years, there's a lot of genetic and molecular tools that have been developed that can be optimized and were used in mice. So these tools that the previous Narset Awardi Carl Deisser developed to activate and inhibit cells was optimized to use in mice, for example. Or in mice, you can also very easily see what's the result of increasing or decreasing the expression of a particular gene or protein product. So we have a lot of kind of genetic access to changing features in mice. So
But
that's
also,
why it's done in mice.
almost shockingly to, I think, people who know nothing about animal research, the mouse brain is astonishingly like the human brain up to a certain degree, but I mean structurally and functionally in many cases. Because evolution has conserved certain properties of the brain in mammals because it was so advantageous. Yes, that's why you can do
Yes.
human relevant research in an animal seemingly as distant from us as a mouse.
Yes, that's completely right. Yeah, the brain is very structurally similar. But as you pointed out, it's not identical in everything. So there are some brain circuits that are more similar to humans than others. And generally, the view is that behaviors that are more species specific are being governed by brain regions that will tend to be more different between these two animals, human and mice. So, for example, like the mouse uses their whiskers to sense the world. So the brain circuits for whisker sensing are kind of don't have an equivalent in humans because that's a species-specific action. But this, which is seeking fatty foods and such, is very similar to what humans are doing. So this would be expected to be more similar in humans and mice compared to like whisker and representing brain regions. And to the extent we can tell from the studies in humans, this brain region, the periaqueductal gray, does seem to have a similar action because in both animals, when you activate all of the cells in this brain region, it seems to cause a very rapid panic fear response in rat, cat, or human.
Well, actually, I'm glad you mentioned that because that was my next question, my next to last question, in fact. Uh, Much of your research prior to this, although you were looking at the periaqueductal gray, the PAG area at the top of the brainstem, this small area,
Yes.
it's really important in the flight or fight response, in the response to fear. Uh, and you were doing basic research on this area and have had a lot of findings that are relevant to fear and anxiety
Yes.
and have nothing to do with this. And yet by stimulating these cells in these inhibitory cells, this group of inhibitory cells within this area of the PAG, you discovered something about ultimately perhaps about reward. Uh,
Yes.
this is remarkable, and I think it's such a great illustration of why the government of this country has made such a commitment to basic research. You had no idea that you were going to come up with anything that had to do with eating behavior because you weren't really thinking about that. You were thinking about PTSD, anxiety, maybe depression, certainly the panic attacks, but not eating and reward. So this is a great illustration of basic research. Yes, you were quite.
Yes, definitely. Yeah, it's both surprising and also it turned out to be much more interesting and important than if we just had verified our initial hypothesis. Because if we just verified what we had predicted, which is, oh, these cells inhibit fear, then that would be a finding for that field that was somewhat expected and predicted. But this finding is completely unexpected. Like the reward studying people didn't think this would happen, or the alcoholism addiction people also wouldn't expect anything to happen here. So it's a yeah, completely different result and much more fruitful and interesting than if our hypothesis had turned out to be correct initially. Yeah, so it really illustrates the importance of doing basic research. I agree.
Right, and that's why BBRF funds it as well. One final question, just where is this all leading you in the next couple of years, up to 5 years, perhaps? It might not be reasonable to ask beyond that. What are you doing now and in the near future? Where is all this leading you?
Yes, excellent question. So now we are studying how these cells promote seeking of other types of rewards that are not these food caloric rewards, such as potentially drugs of abuse like alcohol or social interaction, which is a completely different type of reward that this also seems to engage in. And we also want to see if the cells that represent those different rewarding aspects and the deprivation of those stimuli are the same. So here we see that there are some cells that are more active in food deprivation, which is hunger. So we want to see if you have social interaction deprivation, will the same cells that are active during food deprivation are also active during social deprivation? And if so, that would suggest that these cells are really encoding some kind of internal desire for some external reward rather than some food specialized cells and some social interaction specialized cells. And are those also the same cells that get activated when there's alcohol deprivation in the mouse that has learned to binge drink? So we can see if these are separate or unified circuits within this population. And because the cells are geared towards reward seeking, we can also investigate if their function is tied towards disorders that are known to involve pathologies of motivation and reward seeking, such as in depression, where there is less reward seeking, less motivation to seek pleasurable activities, or in addiction, which has sort of the opposite result of kind of a pathological focus on one type of addictive reward seeking.
Avi, I have to stop you there, and I'm sorry. It's very interesting, and we could continue on, but we're going to have to stop there. I just want to thank Dr. Avi Adhikari once again for providing us with a really enlightening presentation, and I'd like to thank the audience for joining us. I would also like to ask each of you to please consider making a donation to BBRF today. 100% of every dollar donated for research is invested in our research grants, and we can do this because our operating expenses are covered by separate foundation grants. And this means that when you donate $1 for research, that dollar goes directly to the scientists. So to make a gift, please visit bbrfoundation.org or call 800-829-8229. This webinar has been recorded, and if you missed any portion of the presentation or would like to share it, please visit the events and webinars page on the BBRF website. Please join us again next month when we present another mental health research webinar on the 14th of July at 2 p.m. Eastern Time, and we look forward to seeing you then. In thanking you, I ask that you remember the sage advice that our regular host, BBRF CEO Jeff Borenstein, offers each time we meet. He says, together we can dramatically improve the lives of those living with mental illness and enable more people to live full, happy, and productive lives. So with that, I thank you very much for joining us today. Bye-bye.
Thank you.